In the crushing darkness, 2,000 feet beneath the waves, something impossible moves through the void. It stretches longer than a blue whale, yet weighs almost nothing. It is not one creature, but thousands living as one. It has no brain, yet hunts with calculated precision. It glows with a red light that shouldn't exist in these depths, twitching lures that mimic the dying throes of prey. This is the Siphonophore, and what scientists discovered about it in April 2020 would challenge everything we thought we knew about life in Earth's deepest waters. When the research vessel Falkor deployed its submersible into the Ningaloo Canyons off Western Australia that spring morning, the team expected to catalog new species. What they found hovering in the water column at 625 meters depth made them abandon their planned route and flood into the control room. On their screens, coiled in a perfect spiral like some alien spacecraft, was a creature measuring 150 feet from end to end. Scientists later calculated it might be the longest animal ever recorded. Dr. Nerida Wilson, staring at the footage, described it as a spiral UFO hovering in the midnight zone. But this was no visitor from space. This was something far stranger, something that had been perfecting its design in Earth's oceans for over 540 million years. To understand the Siphonophore is to reimagine what it means to be alive. We think of organisms as individuals, distinct bodies with organs working in concert, but the creature that hung suspended in that Australian canyon was neither singular nor plural in any way we can easily comprehend. Every inch of its translucent rope-like body was composed of thousands of tiny beings called zooids, each one a clone of the others, each one incapable of surviving alone, yet together forming something that moves, hunts, and thrives in one of Earth's most hostile environments. The story of this living string begins in the ancient seas, in an epoch when Earth's oceans were just learning to support complex life. The Cnidarians, the phylum that includes jellyfish, corals, sea anemones, and Siphonophores, emerged roughly 580 million years ago in the Precambrian era. They're among the oldest animal groups on our planet, predating the explosion of life that would define the Cambrian period. While most cnidarians evolved into relatively simple forms, some jellies pulsing through currents, others cementing themselves to rocks as corals, the ancestors of Siphonophores took a radically different path. By 540 million years ago, the class Hydrozoa had appeared in the fossil record. Within this group, something extraordinary occurred, something so unusual that it represents one of evolution's most perplexing innovations. Individual polyps, tiny jelly-like creatures, began to remain connected after budding from one another. Instead of separating into independent lives, they stayed linked, forming chains. Over millions of years, these chains grew longer, and the individual zooids began to specialize, each taking on a specific role within the colony. Some developed into swimming bells, pulsing with rhythmic contractions to propel the chain through water. Others transformed into feeding polyps, extending long tentacles armed with microscopic harpoons. Still others became reproductive organs, or gas-filled floats, or protective bracts. What emerged was neither a single organism nor a simple colony, but something in between, a superorganism that blurred the boundaries of biological classification. The Siphonophore represented a kind of evolutionary cooperation so complete that the individual zooids lost their ability to exist independently. They became organs in a body that had no central nervous system, no brain to coordinate their actions, yet moved with the unity of purpose we associate with conscious thought. And then, mysteriously, they vanished from the fossil record entirely. Despite having existed for hundreds of millions of years, despite thriving in oceans across the globe, Siphonophores left behind no stone impression of their existence, no trace in ancient rock. Their bodies, composed almost entirely of water and held together by pressure and a thin gelatinous tissue, simply dissolved when they died, leaving future scientists with a profound absence where evidence should be. We know they survived mass extinctions that obliterated entire orders of life. We know they adapted and diversified while continents split and reformed. But how they did so remains locked in a past that left no record. What we can observe, however, is the result of that deep history, the creature that exists today in Earth's oceans. And nothing prepares you for the reality of encountering a giant siphonophore in its natural environment. Imagine descending into the ocean's twilight zone, where the last remnants of sunlight fade into absolute darkness. The pressure at these depths, 700 to 1,000 meters below the surface, is crushing, between 46 and 70 atmospheres pressing against every surface. For creatures with rigid skeletons, this is a death sentence. But the siphonophore has no bones, no hard structures at all. Its body is 95% water, held in shape by hydrostatic pressure. It doesn't fight the ocean's weight, it becomes one with it. In this realm of permanent midnight, where temperatures hover just above freezing and food drifts by in sparse, unpredictable intervals, the siphonophore floats like a transparent ghost. Its body is translucent, nearly invisible, an adaptation that renders it almost undetectable to both predators and prey. Imagine a living string, as thin as a broomstick but longer than a blue whale, drifting through darkness so complete that your eyes, no matter how long you waited, would never adjust. This is the siphonophore's domain, and it has evolved into a perfect expression of efficiency in an environment that tolerates no waste. The colony's structure is a marvel of biological engineering. At one end, depending on the species, floats a small gas-filled organ called a pneumatophore, like a microscopic balloon that provides buoyancy. Just below it cluster the nectophores, bell-shaped swimming zooids that pulse in rhythmic contractions to propel the chain through water. The smaller nectophores near the top handle steering and orientation, while the larger ones at the base provide the main thrust. This distributed system means that if some swimming bells are damaged, others compensate seamlessly. There is no central command structure, no command string, yet the colony moves with fluid grace through three-dimensional space. Trailing behind this locomotive system stretches the stem, a long central column from which hang the specialized feeding zooids called gastrozoids. Each gastrozoid extends a single tentacle into the darkness, and from each tentacle branch dozens of tentilla, tiny side branches studded with nematocysts, stinging cells that contain some of the most sophisticated venom delivery systems in nature. When triggered by contact, these microscopic capsules explode in less than a millisecond, firing hollow harpoons that simultaneously anchor prey and inject a cocktail of neurotoxins that paralyze victims almost instantly. The speed of this discharge generates forces over 5 million times the acceleration of gravity, making it one of the fastest cellular processes known to science. But in the deep sea, where prey animals are few and far behind, simply waiting for something to brush against your tentacles is an inefficient strategy. This is where the siphonophore reveals its most haunting adaptation. Many species of deep-sea siphonophores are bioluminescent, capable of producing light through chemical reactions within specialized cells. Most emit blue or green wavelengths, colors that travel farthest through seawater. But some members of the genus Irena do something no other marine invertebrate was known to do until scientists discovered them hunting off the coast of California. They glow red. Red light at these depths is revolutionary. The short wavelengths of red don't travel far in water, which is precisely why most animals can't see them. But Irena doesn't need the light to travel far, it needs it to deceive.
On each tentillum, specialized organs produce bioluminescence that fluoresces bright crimson, and these glowing tips twitch and flick in a pattern that perfectly mimics the movement of small copepods, tiny crustaceans that form a crucial part of the deep-sea food chain. To a fish hunting with its own bioluminescent headlights, these twitching red spots look exactly like an easy meal, the red glow similar to that produced by algae in a copepod's translucent gut. The fish approaches, the lure flicks, the fish draws closer, and then, in darkness so absolute that the difference between life and death is measured in millimeters, the fish touches the nearly invisible tentacles surrounding the glowing bait. The nematocysts fire, the toxins flood the prey's nervous system, and the tentacle contracts, reeling the paralyzed fish toward the gastrozoid's mouth, where it will be digested and its nutrients distributed through a vast canal system to feed every zooid in the colony. This is cooperative hunting taken to extreme. The zooids that produce the lure cannot digest food. The zooids that digest food cannot swim. The zooids that swim cannot reproduce, yet together sharing all nutrients through their connected